Now, when it comes to acquiring antibiotic resistance, there's two ways under which this is done, which is through either mutation or horizontal gene transfer. Now, in terms of mutation, there are many different ways that this happens, and one of them being an actual mutation within the DNA. So, what happens here exactly is a mutation, which is a change in the DNA, what can cause a gene product to change, which will allow it to not do exactly what it's trying to do. So, take for example, in this scenario, we have a DNA gyrase here, which is replicating the DNA, and let's say this orange thing is an antimicrobial. Now, this would come in and basically inhibit the DNA gyrase's ability to replicate this DNA, stopping the DNA replication, and would stop the growth of that DNA as a whole, the replication of that DNA as a whole anyway. Now, what happens with a mutation is whenever these spontaneous mutations occur in specific areas of the genes that are encoding these enzymes, these mutations will do a change, let's say, such as this, where that structure change a little. So now, whenever this antimicrobial comes to block the DNA gyrase, it no longer binds efficiently because it no longer fits in that pocket, which allows the bacterium to continue DNA replication, leaving it antibiotically resistant. Now, the gene of interest in this scenario, or the resistant gene, would be not really a specific gene, but the fact that these mutations are happening, allowing the cell to stay alive. Now, this can also be shown in destruction inactivation, where basically we have another cell here, or another bacteria cell, and what's happening here is it's producing enzymes that chemically degrade or deactivate antimicrobial, which leaves them ineffective against the bacteria. So let's say this is an enzyme that destroys or inactivates any microbials that come in. So whenever this group of antimicrobials come into the cell, this enzyme goes to work going at each of them and destroying or inactivating them, overall completely getting rid of their effectiveness and getting rid of them as a whole. This would just not let the enzymatic this would not let the antimicrobial reach the target site and damage the bacteria cell, but before the enzyme obviously gets to it, it's destroying and inactivating it, so there's no growth or accumulation there. Now, this same concept is here in the efflux pump, where basically, once again, we have a group of antimicrobials coming in here. They come through the porins of the cell, and whenever they come in, this thing right here, which is called an efflux pump, immediately is at work and immediately shoots these out of the cell before it can have any effect or accumulate at all and cause any type of damage. Now, this channel actively exports antimicrobials and other compounds, and prevents intracellular accumulations to exert their lethal activity inside of the cell. Now, those are all types of mutations and ways that they stop the effectiveness of antimicrobials. But another way of acquiring antibiotic resistance is through horizontal gene transfer. Now, horizontal gene transfer relies heavily on one cell taking this gene of interest or resistant gene to the other cells. So in all of the examples we have, we will be using this purple as the example of the gene of interest. So let me zoom in here for a second. And as you can see, we have in conjugation, we have this cell with the obvious gene of interest, the resistant genes, while the other cell does not have this gene of resistance or resistant gene. Yeah. So basically what happens here is whenever these two cells come in close proximity with each other, this thing called a pilus will form. And basically, a pilus is just from the pili of the from the pili of the two cells whenever they're super close together. But these two would come really close together, and a pilus would form like this right between the two cells. It would form like a kind of hollow bridge-like structure. Now this allows a copy of the plasmid to, like let's say this plasmid, which has the gene of interest, would be copied so that it still has its own copy and is making this copy, and then transferred through the pilus to the other cell, where this cell would now have that gene of resistance, or that gene of interest. Sorry, I keep mixing it up. That gene of interest, or that resistant gene. Now, this enables susceptible bacteria to acquire resistance to a particular antimicrobial agent without involving cell death, as we will see in these other two examples. Now, in these other two examples, let's start with transformation. Basically, what happens here is it's kind of similar to conjugation, except the fact that this cell will end up dying. So we start here with just a cell with the gene of interest, gene of resistance, whatever the gene of interest, the resistant gene. And what's going to happen is whenever this cell undergoes cell death, or whenever it dies, these parts will be free floating around, and we can even see that through these parts will be around. It'll just be kind of free floating out in like the area around the cells. So now other cells that are nearby, such as this cell, will incorporate these into its own DNA, like so. And after doing that, it'll take in all of these different things, and that would cause for it to gain these new genes of interest, these new genes with the resistant gene, the favorable gene on them, through cell death. And just to reiterate, other bacteria in close proximity scavenge this free-floating DNA and incorporate it into their own, and it may contain advantage genes such as the antimicrobial resistant genes that we see with those purple parts on there. And finally, we have transduction, which is whenever bacterial DNA is transferred from one bacterium to another inside a virus that infects bacteria. So these viruses are called bacteriophages, or phages, I think you say them. They would be these guys right here, these cool looking guys. And basically what happens is when a phage infects a bacterium, it essentially takes over the bacteria's genetic process to produce more phage. So whenever these phages take over the cell, it'll go ahead and start taking over like this bacterial cell's DNA. And whenever it does this, you'll see that there are resistant genes on these bacterial cells, and these resistant genes will begin to go away into the these resistant genes will begin to go away into the actual viruses. I'm trying to move this really quickly to demonstrate. But as you can see, and let me zoom in really quickly just to make this a little easier myself. You can see these resistant genes will now go onto these bacteriophages, showing that they have this resistant gene on them now. So what happens is whenever these cell ends up dying or lysis occurs, these phages will go to infect new bacteria because they need to be infecting other bacteria to survive. So this cell will die, and they will go and leave, and they would actually take these with them, but they would group. let me group this really quickly. They would take these genes with them and go to this other cell on the side here. And this, whenever they come to this next cell with these resistant genes, they would spread these genes that are inside of their own like DNA inside of their capsules with the gene there with the bacterial cell that they are infecting, therefore causing these genes here to turn into these genes with the genes of interest on them, or the resistant genes. Therefore, sharing the gene of interest through the vector of a bacteriophage. So just to reiterate, overall, let me zoom this out really quickly. You can see that we have many types of way that the bacterial cells have antibiotic resistance, and that they can cause for the antimicrobials to not be as effective, which ends up generating these superbugs. You can see that in our mutations, with our mutation, our destruction and activation, and our efflux, that are all working to stop the antimicrobial's effectiveness, or completely inactivated or booted out of the cell. And you can also see that through the horizontal gene transfer, where they're taking these genes of interest that we see on the plasmid, which are on these circular DNA pieces, and they are either giving these to their other cells through conjugation, where it goes through a pilus, or through transformation, where a cell dies and the naked free-floating DNA is spread to the other ones, or through transduction, where through a medium such as a bacteriophage is how that resistant gene gets spread. That is overall the ways of acquiring antibiotic resistance. Thank you for listening, and I hope you enjoyed.